So I'm Helen Wilsey. I'm an assistant professor at UCSF San Francisco, and I'm also an investigator with the Zuckerberg BioHub. And today I'm going to give you some updates of what we've been doing on DYRK1A, and particularly its role at the cilium, and then also in these gut issues that are so prevalent. My lab at UCSF really focuses on autism spectrum disorder genes. So this is a snapshot of the current view of autism genetics, where there are common variants of individually very small effect, and then rare variants of individually very large effects, increasing relative risk over 100-fold. They're very rare variants, and so we like to study these genes to start to understand how they work together, and then to unfold the underlying biology of autism spectrum disorder. So that's how I got interested in DYRK1A. You'll see it's one of these high confidence, large effect risk genes for autism spectrum disorder. Now, on any given day, my lab is usually studying about 30 to 50 of these genes. So in order to do that, my lab uses a high throughput frog model. So let me tell you why we're doing frogs. And specifically, I'm talking about Xenopus tropicalis. Historically, people have used Xenopus laevis, but they, like fish, have a duplicated genome, so it makes the genetics a bit challenging. Instead, Xenopus tropicalis has a diploid genome, so one copy from mom, one copy from dad, just like humans. It makes the genetics a bit more straightforward. One pair of frogs will lay over 4,000 embryos in a day. This is a 15-centimeter dish. Each one of these little dots is a fertilized embryo that we can do experiments on. They have rapid development, so in under a week, you can get a free swimming, behaving, eating, pooping, you name it, tadpole, and have a full organism within a week. They're cost effective, so my animal costs are orders of magnitude cheaper than a mouse lab, and that enables me to study many different genes all in parallel. They're really great for drug screening, and I'll show you a little bit of that effort today in DYRK1A. Because they are developing outside the mother and in an aquatic environment, we just add the drugs to the water and they go into the embryo, and we can see what effect that drug has on the embryo. So it's scalable. We can do it in 96-well plates. So it's a nice platform also for drug screening. But my favorite thing about frogs, and if you remember one thing about frogs, please let it be this, is some cool tricks we can do with genetic manipulation. So we can actually make half-mutant animals. So if we catch the embryo right after that first cell division of life and inject CRISPR-Cas9 reagents with a dye into two cells, that cell ends up generating the whole right half of the organism, and the left cell, which is unmanipulated, goes to contribute to the entire left half of the organism, thereby creating an animal where you have an internal control within the animal itself. So we think this is really powerful to do large-scale genetics, to have an internal control within the animal, and again, it's scalable. It's only about $25 per gene. And this is a typical afternoon in my lab where we have hundreds, if not thousands, of half-mutant embryos that we're creating, and to understand gene function with a bit more detail. You may be saying that humans and frogs are pretty different, but I'll tell you, we're pretty similar to frogs. If you take the DYRK1A protein sequence and align it with the mouse and the human version of DYRK1A, you'll see that large stretches of the protein are completely identically conserved. But actually, there's a few places where the frog sequence is actually closer to the human one than the mouse one is. But these boxes here are showing you the individual differences in amino acid identity comparing frog and human. So this protein is highly conserved. And so we think that we find in frog could shed light on functions in the human as well. The first thing we did when we started studying DYRK1A, it's a classic frog experiment, is you figure out where the gene is on. So this is an RNA in situ hybridization against DYRK1A in the early gastrulating embryo, a neurulating embryo, a tailbud embryo during organogenesis, and then into the tadpole stages where the animals are now behaving and swimming. One thing kind of popped out to us was that Dirkwinney was really highly expressed in tissue that really rely on a subcellular structure called the cilium. And so in here is the epidermis, the skin of the frog, which is actually highly ciliated in a punctate pattern. We could kind of pick that up here. We said, it's really high in the ciliated epidermis. It's also really highly expressed in the brain, particularly in the neural progenitor cells, the kidney, which is a highly ciliated tissue, and the otic vesicle, again, very highly ciliated. Then when we looked at where the protein was localizing, we looked again at this epidermis cells, and we could actually find DYRK1A by antibody staining, and we validated this antibody in Xenopus. And you could actually see it localizing in a punctate pattern along ciliary axonemes. So don't worry, I'm going to tell you what cilia are in a minute. Okay. So you see it also in the cell membrane and then the ciliary axonemes. Again, starting to give us this picture that DYRK1A may have functions on the structure called cilia. So cilia are and why we should care about them. So cilia are important for almost every cell in the body. They come in two flavors. Cells either have motile cilia, and so this is one cell that puts up 150 to 200 cilia. This is on the frog epidermis, and they actually function to move mucus and to create fluid flow. So they're actually hanging out, just beating like you would not imagine. And so on the frog skin, they're moving mucus and creating a barrier against pathogens. But this same kind of thing is happening in the respiratory tract, moving mucus, and then also in the ventricles of the brain, moving cerebrospinal fluid. A sperm tail is a motile cilium, and a lot of the reproductive organs in females have motile cilia to move the follicle, and other things like that are motile cilia. Almost all the other cells then will have a non-motile cilia. So this is a human neural progenitor cell in culture. And you can see the small cilium right there in pink is called a primary cilium. And so this structure is especially important in neurons and in brain development because it controls all of sonic hedgehog signaling, a major player in embryonic patterning and the distinction between excitatory and inhibitory neurons in the early embryo. It's also a big site for regulating excitability of neurons, and as well as there are specific neuronal signaling receptors that are exclusively found on the cilium. So for example, somatostatin receptor 3 is localized to the cilium. Dopamine receptor D1 is specifically localized to the cilium. And many different serotonin receptors are specifically localized to the cilium in neurons. So we think that this is an understudied but exciting organelle to be thinking about neuronal signaling, neuronal excitability, and potential downstream effects in patients. While the cilia field is still a bit in its infancy, actually cilia were thought to be vestigial structures until the 1990s when we realized they're very important for kidney development, and then we figured out they're very important for heart development, and then we figured out it's very important for brain development. So there's now this nice field studying ciliary biology and understanding what goes wrong when there's a cilia defect. So when there is a cilia defect in a human, it will cause a multi-organ spectrum of symptomatology, depending, again, if it affects the motile cilia, if it affects the primary cilia, if it affects both, which tissues that gene is expressed in and required for, etc. But some of the common presentations of a cilia defect include epilepsy, intellectual disability, hydrocephalus, congenital heart defects, chronic respiratory problems, kidney issues, skeletal anomalies, craniofacial differences, infertility, hypogonadism, and more. So again, if there is a cilia defect, there is then this multi-organ spectrum of effects. So during my postdoc, I wanted to develop this idea a little bit more with DYRK1A. We saw that it localized to the cilia on, but does it do anything there? So we took two complementary approaches, antisense oligonucleotides, or AKA morpholinos, and knock down DYRK1A there, or knock down DYRK1A with a CRISPR. And so this is that frog skin where you have these individual cells that each are putting up 250 cilia. Those are the pink are the cilia, and that's an individual cell there with all of its cilia. But when you knock down DYRK1A, these cells have trouble forming, they have trouble making cilia, both in the morpholino and in the CRISPR conditions. So it does look like DYRK1A is required for cilia formation in this tissue. And there's been more work now in frogs and other models to understand why this is. And so I think there's two really relevant things to why DYRK1A might be important for cilia. The first came back from the Jan lab in 2016 when they showed that DYRK1A in the homolog and Drosophila mini brain actually directly localizes to microtubules, which are the main component of cilia. And that DYRK1A actually directly phosphorylates microtubules to regulate its stability. So microtubule stability is really important for forming a cilium, since that is the main structural component of cilia. And so something that is affecting tubulin stability will certainly oftentimes cause a cilia defect. The other work that's been done and that I think is particularly relevant is from Ira Dar's lab, who has built on this work now to show that DYRK1A during the early steps of ciliogenesis actually phosphorylates CEP97, which is a key player in regulating the formation of these basal body structures at the base of cilia. So I think these are probably two of the most relevant mechanisms to why loss of DYRK1A ends up causing cilia defects. But everything I showed you was frogs. So the next thing we wanted to do is see if we could see any evidence of ciliary dysfunction in humans with DYRK1A variants. So again, we borrowed from the ciliopathy field, where a classic biomarker for a cilia defect has been nasal nitric oxide. So what this is showing you here is that in the nasal passage, there are these multiciliated motile cilia that are beating, just like I showed you with the frog skin at the beginning, that are just constantly beating, moving mucus. If those cilia are present and they are beating properly, then they will produce nasal nitric oxide or nitric oxide from the nasal passage. However, if there's a cilia defect, either structural or mechanical, and those cilia cannot beat, much less nitric oxide is produced. And so this is now a highly sensitive, highly specific biomarker for diagnosing primary ciliary dyskinesia, a classic ciliopathy. So we wanted to say, could we use this as a potential biomarker or informative metric to be thinking about DYRK1A and other genes that I study in cilia biology? So it turns out you can buy a portable nasal nitric oxide machine. It's about the size of a Kindle. It's made by a company named Niox, and it's a 30-second non-invasive test, and we do the tidal breathing method. So all you have to do is put this nasal olive superficially into the nose of the person you want to measure, and they just breathe in their mouth normally, and 30 seconds later, it will give you the reading of the amount of nitric oxide produced by the nose. We like this test a lot because, again, non-invasive, 30 seconds. We've done over 200 people now, and of those 200, and with a lot of kids with intellectual disabilities and behavioral challenges, only 2 of those 200 were unable to complete the test. Okay, what does it look like for DYRK1A? So again, we always compare the individual with a DYRK1A variant to an unaffected family member, either a parent or sibling. And then we can test nitric oxide of the unaffected family member compared to the individual with a DYRK1A variant. And here we can see a statistically significant drop in nasal nitric oxide for individuals with a DYRK1A variant. Some of these are actually even low enough to meet the diagnostic threshold for primary ciliary dyskinesia. And so we think that this is an exciting finding that we are trying to expand now and follow up on. And this effect is significant even, it's not affected by age or by sex matching. So we checked all those things. Okay. So far what I've been telling you about has been motile cilia. But in the frog, we have the opportunity to study many different organ systems. And like I told you, those primary cilia are really important for the development of many different organ systems. So I'm briefly going to tell you a little bit about the primary cilium's role in the organization of some of these other organs. All right, so I showed you motile cilia before, but just like motile cilia, DYRK1A here is also localized to the primary cilium. This is actually in an enteric neuron in the gut, and I'll talk more about those cells in a minute. But then also, and so this is frog, but they're also DYRK1A is really beautifully localized to primary cilia and retinal pigmented epithelial cells, which is a classic cell model in humans for studying ciliary biology, because they make these beautiful long cilia and they're really easy to work with. It's an immortalized cell line. But when we use a DYRK1A antibody that we have validated through loss of function, again, we see this beautiful localization with a cilia marker, RL13B, on these human cells. So we don't think that this is a motile cilia specific effect. We think that the primary cilia also could be impacted by DYRK1A loss of function. So let's talk about those other organs. So one of the things we saw early on with DYRK1A was that it was really highly expressed in the pronephros, which ends up becoming the kidney of the frog. And so Rachel Miller's lab in collaboration studied the role of DYRK1A in the developing kidney of the frog. And so that's normally what the kidney looks like. But when you knock down DYRK1A, you get this kind of cystic and poorly formed kidney. And then when she adds back DYRK1A, human DYRK1A, you can rescue the formation of the kidney. And then when she uses a patient-derived variant, that does not rescue. So this really, DYRK1A, at least in frogs, has a really important role for kidney formation. And then when they actually went and looked at patient populations, they could actually find quite a few renal or kidney UTIs among the patient population that they were looking at in DYRK1A. So it seems that DYRK1A definitely has a role in kidney development in frogs, and there are some echoes of this in human patient populations. So kidney, let's talk about the brain. So we also saw that DYRK1A was highly expressed in the developing brain, especially around the ventricle where the neural progenitors are. And so what we did, now we're looking at a frog brain. So we do our cute trick where we mutate DYRK1A just on the right half of the embryo. And so the left half is your control. This is the telencephalon region of the frog. And you can see a reduction in the overall size of the telencephalon when DYRK1A is mutated. Interestingly, when we overexpress the human version of DYRK1A, the brain gets bigger. You put them together and brain size becomes restored. We also, in this paper, did a drug screen. I'm not going to talk about it, but we did a drug screen against this phenotype here. So this is a control forebrain. These green cells are the neural progenitors. The pink cells are the differentiated neurons. When we added a DYRK1A inhibitor harmine, we saw a massive sort of overrepresentation of neural progenitor cells compared to differentiated neurons. But we did a drug screen that then found estrogen could rescue this ratio, and we figured out that estrogen was actually functioning through the sonic hedgehog pathway. Cyclopamine is a sonic hedgehog inhibitor, and that could actually rescue it. So again, sonic hedgehog signaling is coming up, which of course only happens on the cilium. So again, this is an interesting finding that sonic hedgehog signaling can be manipulated to be able to restore development of the brain. Okay. For the rest of today, I'm going to talk about the gut. And it was really great to see the prevalence of gut issues in DYRK1A being one of the strongest things that are reported, right? And I actually only started this project because of a DYRK1A Foundation Zoom we did in like 2020, where parents were telling me, yeah, but what about the gut? Like, that is a really big problem in our community. And I said, oh, it's a good use of frogs. And so again, I don't just study a lot of genes. And so we kind of broadened this out from DYRK1A to a more general question of why do autism and gastrointestinal distress co-occur? So not just DYRK1A with the gastrointestinal issues, seems to be a lot of autism in general has issues with gastrointestinal issues. And so here's Kate McCluskey. She was the graduate student who really led all of this work. You can see our poop emojis. She recently just defended her thesis and published these findings. And so she's now Dr. Kate McCluskey, but she's sticking around for a little while to do a postdoc to continue some of this work. And so, yeah, a lot of what I'm going to show you, though not all of it, is in this publication now in Nature Communications. So check it out. I'll show the QR code again at the end. So the first thing Kate wanted to do was get a sense of the prevalence of these GI issues among these rare disease genes. And so we use the Simons Searchlight survey data. So thanks to the families that filled out the Simons survey. And so you can see what we just did was take the percent of people with GI symptoms, and we compared someone that has a variant in the gene compared to this population here, which is an unaffected family member. So you can't really see these gray bars because the unaffected family members really don't present with severe GI issues, but you'll see, like you saw in Ollie's presentation, DYRK1A has a very high prevalence of GI issues while the unaffected family members don't report it. For some of these genes, I had access to citizen health medical record data. So this is my plug to get DYRK1A into citizen. I really like this data because it will pull all medical records from an individual and give me diagnoses that maybe wouldn't show up in a caregiver survey. So for example, we see a lot of polydactyly, which is like an extra digit, right? And so for me as a developmental biologist, that's a really big sonic hedgehog signaling symptom, while a patient caregiver survey will never talk about an extra digit, those sorts of things. And then like talking to parents is also really great too, because then they tell me about like their children's odd toenails, and that's very helpful for me actually. And so I like the medical record data because it will have all sorts of things like that. It will have heart murmur, which is a structural heart defect, which is very informative for me. Anyways, so for some of these genes, we have medical record data, so we could start to get a bit more granular with exactly what kind of GI symptom it is, right? In Simons Searchlight, it's very broad, like GI, yes or no. Whereas in the medical record, we can actually look at dysmotility. So actually problems in gut transit. And what we found was for all these genes, they had very high percentage of the patients that had medical record diagnosis codes related to GI dysmotility. And then we could break it down even further into what kind of dysmotility was most prevalent. And just like DYRK1A, a lot of the times it presents as severe constipation, though some reflex vomiting, diarrhea, and pain as well. So that got us really interested in gut dysmotility and issues with moving the bowels and moving contents through the gut. Gut motility is really controlled by gut neuron activity. And so these neurons here that line the gut will fire, and then those will actually move the contents of the gut through it. And so that got us really interested in gut neurons. So the next thing we did was look at developmental single-cell RNA-seq data sets of the human developing gut. And we could look and see, are there any particular cell types within the human developing gut where autism risk gene expression is enriched? So not just DYRK1A, all autism genes. And what we found was that expression was enriched in the enteric neurons. Enteric neurons are the neurons that are in the gut. As well as their migratory progenitors. So enteric neuron progenitors, also known as neural crest-derived cells, ENCCs here. So both the gut neurons and their migratory progenitors had enrichment of autism gene expression. We then kind of lumped it together to make the test more straightforward. And so we lumped together all the non-ENS cells, those migratory progenitors and the enteric neurons. And again, we saw the enrichment was much higher in those cell types. So again, that got us really excited and interested in those neurons that control gut motility. Okay, that's humans. Let's study this in frogs. So why do I think this is a great use of frogs? Okay, so I already told you some of the really exciting reasons why using frogs is cool. The other thing is that we can actually watch these gut neurons. Can't see it great here, but we can watch these gut neurons migrate into the gut during development. They actually start up near the brain and they migrate into the gut, so we can watch them migrate. We have GCamp reporters where maybe you can see when peristalsis happens, you see the, whoa, there, you can't see it great. There are these little enteric neurons. There you go. Now you can see a few that light up when you can see that there's neuronal firing right when there's peristalsis happening in the gut. Not to bash fish, but zebrafish just have a simple tube structure in their gut, whereas frog tadpoles have a coiled gut that's much more similar to the human. And like I told you before, we love to drug screen in the frog because we could just put the drugs in the water. And so we actually, I'll show you later, we figured out how to feed these tadpoles green fluorescent beads. And then we can actually watch them go through the gut coil and then be secreted out and pooped. Well, that'll come back later. Okay. And so when Kate started this, there actually were no frog models of gut neuron development. So she had to do some foundational work just to characterize the system in general. And so this isn't written on here, but this is PHOX2B in situ of a tailbud stage frog. And these little dots here that started up over here start to come down the embryo to here, and then they start migrating into the gut. So this is the gut region right here. So all these little puncta are developing gut neurons, those migratory progenitors that then get all the way through the gut and are now populating the gut with neuronal subtypes. So you can watch over developmental time to see the migration of these neurons. So Kate took advantage of our cute trick where we can just make 1/2 of the embryo mutant, and then we can compare these gut neurons on the mutated side compared to the internal control within the embryo and say, how did these neurons do? Are there fewer neurons? Are there more neurons? Are they migrating too much? Like, what do they look like? And so then we could make these measurements within an animal, comparing each half of the animal. Before I show you that, I want to remind you that DYRK1A localizes to the primary cilium of these neurons I'm talking about. And what was known for the ciliopathy field, and especially in zebrafish, was that these cilia on those migratory progenitors are really important for the migration of those neural progenitors into the gut. And so we wanted to see if that was true also in frogs. It was well known, some from human work and zebrafish work. And so Kate actually directly showed that when she makes a cilia defect, what you actually see is while on the control side, these neurons end up getting into the gut and populating a lot of it. When you actually have a cilia defect, they have trouble migrating into the gut here. So we were kind of poised to think about gut migration because we knew this is really important for the migration of those neurons into the gut. So what did DYRK1A look like? So here's the control. Again, those are the migratory neural progenitors that become the gut neurons. And so the blue is just showing you what they've colonized, and the black is showing you the whole gut region. And so we can make a measurement of the area of the gut that's been populated. So that's the control. And then in DYRK1A, when we mutate it by CRISPR, you'll see that that area is reduced. It doesn't migrate as far into the gut. We also used a morpholino. Same thing. It's not those neurons are not migrating into the gut, as well as a chemical inhibitor TG003. It's not on here, but we also did harmine, and we see the same effect. So loss of developmental DYRK1A causes issues in gut neuron migration into the gut. And again, this is cool because all these measurements are actually the manipulated side divided by the contralateral within its own animal and then compared to each other. Now, these gut neurons actually form pretty early, like the progenitors are specified pretty early in the embryo. And so we wanted to look a bit earlier now and say, okay, they're not getting there. Are they being made properly to begin with? And so what we found was that they have trouble being made initially. So this is an early embryo. The neural tube is starting to close. And on the outer rim of the neural tube, the neural crust cells, which give rise to all the enteric neurons and labeled by SOX10, are in this population here. And so when we do a control perturbation, these two are the same. Now, when we do a Dirkwinney morpholino or a Dirkwinney CRISPR, what you'll see now is on the injected side, you actually lose the specification of the neural crust there. And Amanda Dickinson's lab has also seen and published similar results as well. So it seems that Dirkwinney is also really important for the early specification of neural crust. That might be interesting to some people as well, because neural crust, and this is cranial neural crust actually, the cranial neural crust goes and actually forms the bones of the facial structures. So a lot of times there will be structural abnormalities and cranial facial features because of underlying neural crust issues. Okay, so it seems that early on, Dirkwinney has a role in specifying the neural crust to begin with. And then later, it also, perhaps has a role in regulating the migration of those neurons into the gut. But we wanted to separate those two, because you could imagine if you're not making enough gut neurons to begin with, like maybe that's the issue rather than actually just being migration. So again, it's a good use of frogs. What we could do was try to uncouple specification from migration. And so we would take an embryo, wild type, unmanipulated embryo, allow the neural crust, these cells, to develop unperturbed. So it'll be like this, the neural crust just fine. And then only when those neural progenitors start to migrate in the gut, we then add a DYRK1A inhibitor right when they start to migrate, and we say, are there still migration issues, even if we add it later? And indeed there are. So here's the control DMSO vehicle treated animal. You can see the population of the gut neurons within the gut is pretty high. But when we use, and we did this with TG003 too, yeah, Harmine or TG003, we see issues in gut neuron migration independent of any function of DYRK1A in specification. This is a positive control. So this is PP1, a RET inhibitor. This is kind of modeling Hirschsprung's disease, where the neurons really don't migrate at all. And so that was our positive control. And then we used meclobamide, which is a monoamine oxidase inhibitor, as an off-target control for Harmine, because Harmine, while it's a DYRK1A inhibitor, sometimes it leaks over into MAO. And so that was our off-target control to control for that. And those animals migrate just fine. And so in this way, we were able to uncouple the effect of DYRK1A on neural crust specification from the effect of DYRK1A in neural crust migration into the gut. So it seems that DYRK1A has both an early and a late role in this cell population during development. Okay, guts. Next, we wanted to actually look at motility of the gut. So we see these issues in migration and the development of those neurons. So once you get to a place where you're actually eating, do you see constipation in this frog model? Do these tadpoles actually have trouble with gut motility? So this was a fun summer where I just said, feed them beads. And so Kate fed tadpoles fluorescent beads for 2 hours. And then you could see the beads within that tadpole gut. And then she could put these tadpoles in 6-well plates, 20 animals per well, let them hang out there for 3 hours. And then she could remove the tadpoles, but leave the excrement. These were termed porta potties that summer. She could remove the porta potties, and then what would be left behind would be the fluorescent beads and all the excrement. And then we could actually just measure the amount of fluorescence per well as a high throughput way to measure gut motility. And so the first thing we wanted to do was see if when we inhibit DYRK1A developmentally, if that ends up causing issues in excretion. And so these are just, these are false colored. Okay, so where the black are, those are the fluorescent beads. And then 3 replicates here just showing you the vehicle DMSO control, the animals poop this amount within the 3-hour excretion portion. However, when they're developmentally treated with this DYRK1A inhibitor, you can see that the amount of fluorescent beads that they are pooping out is reduced. There's that quantification. So next we wanted to be able to see if we could identify any drugs that could come in and manipulate this. So this was called Poopapalooza, which was fun. So we did an in vivo gut motility drug screen. So we took something like 39 FDA-approved drugs that had some sort of either known role in neurotransmission or are known to have gut GI side effects like caffeine or antibiotics, things like that. So we took a bunch of FDA-approved drugs and then on wild-type embryos said, can any of these drugs, and there's the DMSO control. So we just put that there at 0, it's arbitrary. So that's how much a control animal poops, and then we would add a drug and say, does it change the amount of pooping? And then we arbitrarily said anything over 1 standard deviation from that, we're going to call drugs that can increase gut motility, and anything under a standard deviation, those are then drugs that can decrease gut motility. Okay, so for this, we were really interested in things that would increase gut motility. And so 3 of these drugs all hit 5-HT, which is serotonin. And serotonin is known to have a really big role in gut motility, so this all makes sense. And so we said, all right, let's think about serotonin for a little bit and gut neurons, and what do we know about serotonin? This was cool, because it turns out there's a very specific serotonin receptor 6, that specifically localizes to cilia. And so we then were like, all right, maybe there's an underlying cilia defect, and then serotonin signaling is having trouble because there's a cilia problem. Can we manipulate serotonin signaling to compensate for this issue? Yes. So what you'll see here is the control amount of pooping is here at DMSO, and then you add the DYRK1A over development, and you reduce the pooping. Now you do the same thing. You have TG003 during development, but just during that 3-hour pooping time, we added Lexapro, which is a selective serotonin reuptake inhibitor, to just try and neurons with more serotonin and activate serotonergic signaling. So Lexapro was we tried a bunch of different serotonin drugs, but Lexapro was the one that we could find that acute treatment just during the excretion portion of the assay was able to restore gut motility in these animals. And that was, we didn't know that we were going to be able to find one that could help, because there's also craniofacial defects in these frogs that are raised in DYRK1A inhibitor, as you might imagine, because there's neural crest problems. So then the facial structures are different. And so we weren't sure if they could eat enough. Like maybe this is just because then they're not eating enough, right? Maybe that's why there's less pooping. But actually, the fact that we could add the serotonin reuptake inhibitor acutely and then get back to normal pooping levels told us that they were eating. It's just that it wasn't moving through the gut. And so then, so that was just a really broad SSRI, Lexapro. And so then we wanted to go back to this idea that, you know, there's certain serotonin receptors that are specific to cilia. And so that's serotonin receptor 6. So we said, can we modify this receptor and still get the effect? And the answer was yes. So this WEI181187 is a serotonin receptor 6 selective agonist. It's not FDA approved. But even that one was able to rescue some of the pooping. almost to control levels as well. So we're excited about serotonergic signaling and potentially being able to help with gut dysmotility, and specifically serotonin receptor 6 might be a more targeted way to do it rather than flooding serotonin, which of course could have other issues in the central nervous system. Okay, so what I've told you today is that DYRK1A localizes to both primary and motile cilia. It's required for cilia formation, and individuals with DYRK1A variants have lower nasal nitric oxide compared to their unaffected family member. It's well known that cilia defects cause kidney issues, microcephaly, GI issues, and DYRK1A loss of function in the frog causes all of these, with evidence to support it from human patient data. And then we think that manipulating ciliary biology may be therapeutically actionable. The cilium can regenerate throughout the lifespan. So you might imagine that you could actually be able to regenerate the cilium and restore signaling if you're able to affect the cilium. Okay, so what's next? One of the things we're really excited to think about is, when is the therapeutic window? So for DYRK1A, right, we were able to just acutely add this SSRI for 3 hours, and that could rescue, right? So that says the therapeutic window is very long, you know, even long after the developmental issues with gut neuron migration have happened, you can still intervene and restore function. And so now what we want to do is try and restore DYRK1A function at different times during development, and then look at all those things I already showed you: kidney development, brain development, gut function, ciliary biology, and when can you actually rescue these issues, and is it dependent on the tissue type, is it dependent on the cell type, and so what is the therapeutic window? Again, it's a good use of frogs because we can just take out that DYRK1A inhibitor, wash it out at different stages of development, and then see when, if we can restore DYRK1A function, which of these could be actually actionable. We want to drug screen cilia themselves. I showed you that DYRK1A is really beautiful on those human RPE1 cells in culture. Those are really good for drug screening. So what I want to do is drug screen in the RPE1s to actually restore ciliogenesis there and identify drugs that might target cilia, hopefully a little more specifically. We're also interested in cilia phenotyping in humans. I'll tell you that in 1 second. I keep meaning to study seizures, and I haven't done it yet. I don't know if these tadpoles have seizures. I am really also interested in sleep and understanding sort of sleep disruption. Again, good use of frogs. We have automated behavior tracking setups where we can put the tadpoles in 96-well plates, and then with automated imaging analysis, actually look at the activity of these tadpoles, and you can actually see them have circadian rhythm. And unlike mice, which I think are nocturnal, tadpoles are diurnal, so it makes our lives easier. But so we're trying to get into sleep. That's just, yeah. Okay, last thing, if there's any families in the room, we're actively recruiting for 2 IRB approved studies I'm working on. The first is more of this nasal nitric oxide. We got that first group of patients from sending James Schmidt a wonderful postback in the lab to various locations where DYRK1A communities were meeting up. But I think at this meeting here, it'll be really great. I have the machine with me if anyone wants to come look at it and come do this. I just need a unaffected family member as well as the family member with a DYRK1A variant. Anyone over 2 years of age. This is cool. This got approved last week, and I'm really excited about this. And what I really want to do is actually, you can take a brush and brush the back of the nasal cavity and actually scrape some of those cells, plate them, culture them, and they will actually become multiciliated cells with cilia that will beat. And so I want to be able to actually look at the cells themselves from patients with DYRK1A variants compared to their family members and see if we can directly see any issues with cilia formation or with ciliary beating. So please email me. This has to be done in San Francisco. This can be done here or in San Francisco. So let me know. Okay, that's it. So definitely wanted to thank the lab, especially Kate. Poopapalooza was all her. Her manuscript is here if you want to read about it. Yushao long ago started this work as well and laid the foundation for the cilia work. And want to thank our funding sources and our Xenopus specific resources. Thanks.